Hello, this is Pathology and Clinical Science 1. This is Lecture 2, Part 2, and we're looking at abnormal immune response and hypersensitivity. And in this session, we're going to carry on from the last session where we looked at type 1 reactions, and we're going to move straight on to type 2 to type 4 hypersensitivity reactions and looking at understanding the difference between them. So type 2 hypersensitivity reactions are known as cytotoxic. And in this type of hypersensitivity reaction, the antigen-antibody reaction activates complement and products of complement activation. Now this directly or indirectly damages the target cells that are expressing the antigen. So just a little refresher that the complement system plays a critical role in inflammation and defense against some bacterial infections. So complement can be activated during autoimmune disease. In a nutshell, the complement system refers to a series of over 20 proteins which are circulating in the blood and the tissue fluids. And these proteins are normally inactive. However, when various pathogens are recognised by the body, this complement system can be sequentially activated in an enzyme cascade, and this is to eradicate those pathogens. So complement can be acted by different pathways, and the classical pathway is triggered by the complement binding with antigen-antibody complexes. And this can result in opsonization. I can never say that word properly, so apologies for that. This is a process that makes the bacteria more attractive to phagocytes. It makes them tastier. This opsonization happens to microbial pathogens, and it also can cause activation of the mast cells in this classical pathway, and that can lead to histamine release and vasodilation, as we discussed with type 1 reactions. It's that same pathway with that histamine release. So, sorry, I shouldn't say pathway, that's the same type of reaction in the body when histamine is released. Now, the lytic pathway or lytic pathway is also part of the complement system, and this results in lysis or damage to the plasma membranes of the cells and to some bacteria, and it also attracts the macrophages and neutrophils and activates mast cells. The membrane attack complex mentioned here on this slide, it talks about, or it refers to a cluster of specific C proteins from that complement, C meaning complement, that form a structure on the surface of the pathogen cell membranes, and this is following the activation of the complement system. And this is referred to, the MAC is referred to as an effector protein of the immune system. So I just wanted to mention to you that there's a really great page on the website that's the British Society for Immunology. It's a really handy website when you're looking at immune system. They've got great information pages, and there is a really good page about the complement system, and they have some PDFs that you can download and some great diagrams if you want to look into that a bit further, if you feel you're interested in it. So here are some diagrams of type 2 cytotoxic reactions. Now, this diagram A, this is looking at that complement activation that we discussed in the last slide with the opsonization and the MAC and the phagocytosis. Here we're looking at, in diagram B, we're looking at complement and antibody receptor mediated inflammation resulting from recruitment and activation of inflammation-producing leukocytes, or neutrophils and monocytes. And in diagram C, this is an example of what happens in Graves' disease, which is a hyperthyroid, an autoimmune hyperthyroid condition that's due to autoantibodies mimicking TSH. And you can see that there's antibody-mediated cellular dysfunction in which the antibody acts against the thyroid-stimulating hormone. So the TSH receptor increases thyroid hormone production due to this damage. And then diagram D, this is an example of myasthenia gravis, and this is what happens in myasthenia gravis, which is another autoimmune disease. This is where an antibody to the acetylcholine receptor inhibits receptor binding of the neurotransmitter. And in myasthenia gravis, you see very abnormal, severe fatigue of the muscles, and this is caused by autoantibody damage to the acetylcholine receptors at the neuromuscular junction, as is seen in this diagram. Outcomes of type 2 responses: the death of the cell is one possible outcome. Common type 2 reactions are listed here. Now, rhesus incompatibility, this can be treated by using a transfusion. There are a number of different types of drug reactions and, as we've mentioned, autoimmune diseases. So plasmapheresis in this treatment list here is a method of removing a quantity of the plasma from the blood, and then the remaining cells are retransferred back to the host. So moving on to type 3 hypersensitivity complexes. So these are immune complexes, and these are abnormal persistent antibody-antigen complex formation that's not been cleared by those normal mechanisms that the body, that checkpoint that the body normally has to get rid of these antibodies. This is where that fails. So these deposits of the immune complexes in the tissue can cause localized inflammation. They can lead to the vasodilation and edema, and also necrosis or tissue death. So this diagram is looking at these type 3 immune complex reactions, and as you can see here, we're looking at the antibody and antigens, which are forming those antigen-antibody complexes. So they're activating those IgG or IgM immunoglobulins, and in order to form these complexes. Now, these complexes are then deposited in the tissues. It activates the complement system, and this deposition of those complexes in the tissues leads to the attraction of leukocytes, and those are what are responsible for the tissue damage and blood vessel injury, etc. Now let's look at some types of type 3 hypersensitivity reactions. So I'll just give you a little rundown on each of them in case you're not familiar with them. So serum sickness is a reaction that happens generally about 7 to 12 days after an injection of a quantity of foreign serum. Raynaud's phenomena is an idiopathic arterial spasm, and it can cause numbness and pallor and pain and necrosis. Often you see Raynaud's affect hands. Arthus reactions, so there are a number of different ones, as you can see in the brackets there. These are rare, severe, immediate, non-atopic hypersensitivity reactions to the injection of a foreign substance. And that's usually not irritating, but in certain individuals is antigenic. So farmer's lung, this is an occupational lung disease. It's caused by an allergy to fungal spores that are generally found in damp hay, straws, grains. Pigeon's disease, funnily enough, is to do with reactions to feathers, serum, and pigeon excrement. And this is obviously more common in pigeon breeders. And celiac disease, I'm sure most of you are aware of celiac disease. It's very common. I think it's about 1 in 70 people in Australia have it. So this is when the small intestine fails to digest and absorb food due to sensitivity to gliadin, which is a protein that's found in gluten and in a number of other grains. And that is also an autoimmune reaction. So treatment for these sorts of type 3 reactions: corticosteroids, immunosuppressive drugs, removal of environmental triggers, and plasmapheresis. Type 4, cell-mediated or delayed hypersensitivity reactions. Now, remember we discussed that skin patch testing, and this is used to identify type 4 hypersensitivity reactions. And as I had mentioned, that is when you do skin patch testing, use topical application of the allergen over a 48-hour period and then remove it, and then monitor for reaction for 72 and 96 hours following that exposure. So this is an activation of those T cells over time. It's delayed. And really common activators for type 4 hypersensitivity reactions are listed here. So a lot of them are things that will directly come into contact with the skin, Band-Aids and tree or plant irritation from plants, like it says poison ivy here, which is obviously not a very Australian example, but I'm sure there's a number of other plants in Australia that can do some nasty irritation to the skin. Also, dyes, soaps, chemicals, dishwashing detergents, laundry detergents, etc. Oh, and metals. Nickel allergies can be really common. So junk jewellery or cheap jewellery often used to, not so much anymore, but it used to have nickel in it, and a lot of people react to that. And the Allergy Association actually has some information regarding a link between dust mite allergy, nickel sensitivity, and chocolate sensitivity, interestingly enough. And I believe you'll find handouts about that on the it's an Australian, the Australian Society of Immunology, I believe it is, but I have to search that one. Sorry, I don't have that one, that link here for you. Okay, so the mechanism in type 4 hypersensitivity. So these are antigens that are cell or tissue bound. They both involve T cells. The CD4 T cells, remember we discussed CD, cluster of differentiation, CD4 and CD8 T cells, which are the cytotoxic ones. So these are the ones that mediate those delayed cell-mediated hypersensitivity reactions. And here's a nice little diagram for this. So this shows how this antigen comes in. You see the antigen presenting cell. Here are your CD4 reactions and your CD8 cytotoxic reactions. So the CD4s release the cell-killing damaging cytokines, and the CD8 cells kill the antigen-bearing target cells. The clinical features, as you saw in that picture just a couple of slides ago, you can get those vesicles. You can see those blistery looking bumps on the skin. So you often see these in eczema and in herpes simplex breakouts as well. So they're like very small blisters in the skin that contain a clear serous fluid. You can see a localized rash, pruritus, which means itching. You see redness and serous exudate as well. So they can weep, shall we say. Treatment, corticosteroids, removal of the antigen, and in more severe cases, immunosuppressive drugs. And that brings us to the end of this part of the lecture. I'll see you for part 3 shortly.